In this video, we will be discussing about photoelectric transducers. So, what is the working principle of such transducers? It works on the principle of conversion of light energy into electrical energy. When light falls on photosensitive element, electric current is generated that is measured directly or after amplification. What is photoelectric effect? It is the phenomenon of ejection of electrons from a metal or semiconductor surface when illuminated by light or any radiation of suitable wavelength. Okay. What are the types of photoelectric transducers? Photoelectric transducers varies between two things. One is passive transducers, another is active transducers. So passive transducers are those that does not require power source, but active transducers do require power source. Photovoltaic cells. So they're also called barrier layer cells made up of semiconducting substances such as selenium deposited on an ion base. Silver or gold colors this semiconducting material and acts as collecting electrode. Photovoltaic cells or the cells working on the works on the principle of radiating energy upon the semiconductor surface. When it gets excited, it emits the electrons at the silver selenium interface. Okay, the electrons are collected at the collector cathode. If you see in this diagram, you can see that when the radiations light energy falls on the semiconductor surface, at that interface there is a production of electrons in the silver selenium interface, and it's collected in the opposite electrode, that is cathode. Okay. What are the advantages and disadvantages? Photovoltaic cells as robust in construction, need no external power supply. It produces photocurrent stronger than the photosensitive element. Selenium has low internal resistance and the amplification is difficult. Very sensitive galvanometers has to be used for measurement purpose, else it will result in noise. Photoemissive cells. So it consists of a cathode and an anode mounted in a vacuum tube made up of glass. Why glass? Because they can so the light energy can enter. The cathode consists of curved metal plate made up of photosensitive material such as cesium or oxidized silver. The anode is made up of nickel or platinum. When the radiation of frequency above the threshold frequency falls on the cathode, electrons are emitted and flows into the anode, constituting an electric current. The gas-filled photoemissive type cell contains inert gas molecules like argon, which can be ionized. Due to the repeated collision of the electrons, photocurrent produced is greater at even low potentials. So what happens when the light radiation falls? There is an ionization of the inert gas. The inert gas ionization of the inert gas produces electrons, and due to the repeated collision of electrons, photocurrent is produced. Okay. As these electrons are collected in the opposite side of the opposite side charged electrode. Okay. Photoemissive cell types. The photomultiplier tube or the tubes that require to detect very light. So previously we saw that there are phototransducers that will no cannot detect very light intensity, but photomultiplier tubes are constructed for this purpose to detect weak light intensities. Photocathode consists of primary electron diodes, secondary electron diodes, and so on. So when the light radiation falls on these diodes, the electrons are produced, and these electrons are focused on the primary diodes and secondary diodes, resulting in multiplication of this these actually the incoming photon of radiation falls on these diodes, and they get amplified by these diodes, resulting in production of electrodes. Okay, so these electrodes are amplified in a bigger way. So light intensity of minimum value gets amplified because of these diodes, and because of these diodes are produced at because of these diodes, there are electrons produced from these diodes when the photon falls on these diodes, and the production of electrons improves when the light photon increases. The light photons are increased by increasing its intensity, by multiplying the photon energy. So this produces more electrons and more photoelectric current. The operating voltage is 50 to 100 volts. Okay, advantages and disadvantages of the emission is instantaneous. Maximum current is proportional to the intensity of radiation. Increased sensitivity. Disadvantage generates extremely small current. Direct power supply required for photo multiply. Hence, it's called an active transducer. More expensive, can be replaced by silicon diode detectors. Okay, photoconductive cells. So photoconductive cells also called LTR, light dependent resistors. So when light falls, resistance changes, conductivity changes. So disadvantage of photoconductive cells are sluggish and slow hysteresis effects. Okay, photoconductive cells, when the light of the varying index falls on the film, a current flows through the circuit containing a galvanometer and a battery source. So this is also an active transducer. The current directly varies with the intensity of light. So resistance changes, conductivity changes, when there is a photon of energy falls on the selenium silver film. So conductivity of that entire system changes, resistance changes due to the incident radiation. Okay, so biomedical applications or pulse pickups, pneumatogram, respiration, respiratory volume can be measured by measuring the changes in the chest circumference with a pneumatogram, which has photodetector in it. The measured blood pulsatile volume changes, especially in LDR, is used in recording movements of body due to the pumping action of the heart, called ballistocardiograms. So all these measurements in biomedical are used mainly when there is a change in the when the physical parameter changes the incident for photon of radiation. Okay, so this can be used for biomedical applications. And one of the other application is earlobe oximetry. So determination of oxygen in blood is an important parameter, and oxygen of blood directly changes, oxygen of blood, when there is a variation in the oxygen of blood, the incident photon of light or a special wavelength of light, usually in IR light, uh, are sensitive to the changes in the concentration of oxygen in the blood. So this changes in the concentration of oxygen in blood helps in monitoring the amount of O2. So this kind of material is called earlobe oximetry. Why earlobe? Because they are measured at the earlobe. The position of point of measurement is earlobe. Okay, thank you.